biotechnology, bio-nanotechnology, microbiology, and bioinformatics. Among of the line of research that we want to discuss with all of you is our possibility to be involved in the synthesis and engineering of peptides and aptamers by bioinformatics tools. In that regard, our group has a lot of experience designing new kind of proteins and new DNA with high affinity against to any substrate. And also, we will explain with more detail about the identification and characterization of microorganisms capable of biosynthesis of nanoparticles of biotechnological interest. From the next slide, in the number 3, if you can see in your presentation, my colleague Juan Pablo will explain with more detail our progress in that regard.
Thank you for inviting us here today. I am Juan Pablo, a biochemist working on the nanoparticle biosynthesis project. And one type of nanoparticles that I'm going to talk about today are semiconductor quantum dots. And these nanoparticles, well, if you are able to see the slide, here are able to emit fluorescence at different wavelengths. And these fluorescent and optical properties depend on the size of the nanoparticle. Can be better transformational methods because of that they are more environmentally friendly. And they can take place in room temperature. We are currently developing another type, different types of these nanoparticles. Calcium sulfide, like I showed you. Also, different copper nanoparticles, including copper sulfide, and biomimetic and biological synthesis method that forms kind of nanowires of copper that we are now learning to be intrigued about the structures because it's this project to try to feel what enzymes, what molecular mechanisms are promoting the nanoparticle formation. Here, it is very important because this project will be held under the collaboration with Dr. Pastor at San Buffalo and the CBIB Center. This has to promote nanoparticle synthesis apart from other things. And the other thing about that is very good about plastic is that it has low human contamination. There are a lot of signs that no human has ever tested on, so the probability of finding new microorganisms that are not here on the continent is very high. Of all these sites, one of the most interesting is the Central Island, that is kind of in the middle. In the next slide, which is slide number 12, I'll show you a picture of the Central Island. It has an image, and then it goes into a geographical image. Well, this is just to say that it is an island in the South Shetland Islands archipelago. It is one of the latest cargoes in Antarctica because the ship is all the way up there. And the island is actually Aracera, the hottest spot of an active volcano that not settled that much in the 60s. And the island is the first idea that makes us think that the bacteria actually produce these nanostructures in fluorescence when exposed to UV light. And then in the next slide 16, properties and a program of another particle with quantum science. Okay. Well, the last thing, when we discover the microorganisms, we have found some, we need to characterize nanoparticles. All these values are underestimated because actually we don't have the proper treatment to irradiate the cells with actual cell light. It is very standardized because we measure it a lot of times, but these values would be really higher in the cortex equipment. Okay? And well, semiconductor also thought it works as thin film solar cells, another type of dye-sensitive solar cells and batteries, and many others that we haven't really maybe investigated a lot, like that 2071 to now coming soon.
Okay, I will present the next project. Thank you, Juan Pablo. So So the next project is a very ambitious concept, photonic multimode sensor science. The idea is to combine different concepts in the context of to improve the capacity of different kind of systems to detect different applications. The objective of our project is to propose a cross-discipline experimental study and to combine soft material science, laser spectroscopy, nanotechnology. Se está tratando de hacer la conexión, pero okay, well, next slide. I'm sorry, I can't hear you.
No, we can hear you.
Hello?
Hello, yes.
Hello.
Hello.
Okay, I'm good. Okay.
I think he's working.
Potential time? Yeah, okay. That our idea is to make some sensor, new sensor that can detect different explosive DNA virus, and also re-engineering of protein and DNA. And as a first approach, we expect to implement a new kind of sensor that another initiative is in the context of the detection of dengue virus, is that of the main disease that many soldiers suffer in different areas of the world. And for that, we are planning to re-engineer DNA and RNA aptamers in order to improve the detection of different carriers of the dengue to this protein, making different modification in the active site. That strategy is very common in biotechnology, but in that case, our idea is to implement this kind of protein in the context of to detect.
So, Danilo, it's hard with Skype to get a reaction, but it's always a fantastic talk, and I think you explained how well using the unique biology from Antarctica and then the activities within the lab to engineer those is well connects with some of the areas we've been talking about. And also, I want to point out, I guess, to everybody that the lab at Andres Bello is very cross-disciplined, very proactive, world-class, and has been a great part of building these global teams. So, and this, you can see, I think, the work is very good.
Brett, could I ask a question?
And Dr. Chang has another question. I just want to say good things.
I think it's very great. I really enjoyed the short presentation. Now, I got two questions on dengue virus. What are the specific DNA proteins?
It's a Brazil team, also a laboratory experience. They have to do this simulation to the antibody, so they can find this got pretty good. So that's so I think their processing technology is pretty useful for our collaboration. Aeroponic or moving for exposure, whatever.
So I have another question. This is Brett. This is a tough one. So with the idea of multimode sensing, the idea of engineering proteins integrated with optical systems, we talk about dengue fever, we've talked about infrared explosives detection. And this is a brainstorming question, so you may not have an answer, maybe on our return. But can you think of another area or application that's closer to home to Chile than the Chilean Air Force? And I know that's a very general question, but we're looking for the next connection, and we want to be able to find a target, I don't know if that's the right term, an objective that the Chilean Air Force can see the value in more immediately.
Natural contamination, like for example arsenic, that is important to detect in the water also. So I don't know, I think that we need some, I would like to make some meeting with the people related with the Air Force in Chile in order to visualize what is the main problem that they have right now and how we can help you to solve using our technology.
Research center's big challenge facing is IED detection. It's without the metal buried underground. So one way I suggest using the laser shoot there or micro shoot there and a very specific wavelength that the TNT absorb, and then you will generate positive wave, and either positive wave will be detect.
Hyperspectral sensing, spectral sensing in general, especially in IR wavelengths, is very useful for targeting, for any kind of ISR, intelligence, surveillance type application. What this technology could potentially do is bring hyperspectral imaging into a much lower cost intent. That's just one example application.
Excellent, that's great. Any other questions? So, I just want to thank you again. It's a fantastic job and connected well with what the Air Force is doing here, and perfect. Thank you so much. And I will distribute the talk. Sure. And on the return, we're hoping to have some proposals that I want to share with you. So, thank you.
Great. I look forward to seeing those. Thanks for your attention, and sorry about the technical difficulties. I hope some of it came through. It was a pleasure meeting you all, and hopefully we'll get to meet in person someday soon. Thanks again.
Video or telecommunication by using the German base is approximately 10%, but the research indicate that with the associated with nanotube, we can increase the quantum efficiency of the detector to about 2 times.
Use electronic nose to smell many interesting material. I think recently he can smell the jasmine rice. Okay, the smell of jasmine rice. He can differentiate between the regular rice and jasmine rice. Okay, and he also work on simulation, molecular simulation, and synthesis of nanomaterial. Okay, and some intelligent nano device.
This cell that you can see that it lights up under the fluorescence microscope, this is the only cell that was punctured. So we use FIT-C as the dye, and surrounding area was also live cell, but they were not punctured.
Don't worry, my slide will be really quick. I will just go without any detail, just give a broad introduction.
So we have these, we just call research modules. We're very flexible in terms of our mission, in terms of our niche. We're not specific to like laser lab or photonics lab, but we do those sorts of things too. Our projects range from nano-encapsulated fertilizers to electromagnetic modeling of metamaterials. But you can see that the zinc oxide micro rods actually mostly are very disordered and scattered randomly, almost it seems, on the surface of the cotton fiber packaging. So probably most of the pharmaceutical drugs that have the metal on one side and the plastic on the other are probably made by this company. Now, we're definitely inviting MRDC very soon to come visit us. Thank you very much.
We would add, with a proposal from Sitka, the Fatches Research Institute, and MRDC, an element where we would test something here. So the sample would come here under the same